So we learned how omics data can be used to reveal the biological complexity of COVID-19 disease. But how does bioinformatics come into play during this process? And what are some other areas where bioinformatics analysis is used? So now we will hear from Dr. Shuchi to understand the role of bioinformatics in omics study. Dr. Shuchi Nagar is working as an associate professor at D.Y Patil Biotechnology Bioinformatics Institute, Pune. She did her PhD from Department of Chemical Technology, University of Calcutta in the field of cheminformatics and drug design. She has 17 publications to her credit. She has guided around 20 project students. Presently, she has 2 funded projects. One is related to designing anti-cancer drugs, and the other is designing quorum sensing inhibitors. Further, her expertise is also being applied for studying Alzheimer's diseases and for generating Ayurvedic formulation. We are pleased to have you with us, Dr. Shuchi. Over to you. You may share your screen now.
Okay, so before I start off with this topic of relevance of bioinformatics in omics, I would like to just introduce my institute where I'm working. And just to give a brief, the courses that are offered in our institute is we have BTech with biotechnology, BTech with medical biotechnology. We've just started with BTech with artificial intelligence and data science. There's MTech biotechnology and MSc biotechnology. So these are the few courses that are being offered at our institute. And we have different research areas that are there, which are very functional. And there's a lot of research done on these areas. It's microbial diversity. Then you have plant and environmental biotechnology. You have genetics and molecular biology, cancer and translational research, protein biochemistry, and bioinformatics. So these are the 6 broader areas on which all the faculties of our institute are working. And it is we really have a good strength of faculties who are working and doing really well in all these fields. Now, to start with the topic of relevance of bioinformatics in omics, I just want to say that when we talk of omics or when we are talking about bioinformatics, it's like saying that what has come first? Has the hen come first or the egg has come first? That's because it is a part of omics and part of bioinformatics that is common. So you have certain part of omics wherein that part of the omics is completely done on wet lab. Then you have the data that is generated is stored in certain databases. So you collect those data from there, and then you use the different bioinformatics tools to analyze them. So that is why we can, it's very difficult to say whether bioinformatics came first or omics came first. So it's the same story like hen came first or your egg came first. So we can also say it in this way that so it's like, the omics and the bioinformatics are coming together, joining hands to give you a good information that will further help us in systems biology. So systems biology is basically when we are trying to study different pathways that are there in very details. And followed with system biology, we can go to studying or doing cheminformatics studies, where we can go ahead with designing of new molecules, that could be lead molecules for a particular disease of interest or a particular target of interest that we have. So to just understand the flow chart that is there with it, what we actually do is, this is your omics part that is there, wherein we are doing certain kind of your, you can say experimental works. You have n number of different experimental works through which you can get data. So all these could be gene data, genome data, it can be RNA data, it can be protein data, it can be any kind of data depending on what kind of your experiment are you doing. That data, and then we take it and we are storing it in different databases. And then using various algorithms and various tools that are available, we are actually doing all these studies, which is actually the omics studies. Okay. So this is your omics studies, which is your genomics, transcriptomics, proteomics, and metabolomics. So we can say that it's a the follow up that is there is, it's like the central dogma. That's what is there. You have genomics, that is the gene data. So that then you come to transcriptomics, which is your mRNA and other data. From there you have your proteomics, so that's your protein data that you have. Then comes your metabolomics. So ultimately the product that is being formed is what is your metabolomics. So I will lead it. I will discuss on each of these omics in details, as in where can these be applied, and what are the different tools that are available for this. So what are the different kind of studies that you can do with different types of omics data that is available. So definitely you have to have some kind of an experimental work. Only after doing this experimental work can you go ahead and do this study, which is going to be using the data that is generated from here, and then the computer and the various computer algorithms and programmings are used to study the data to get some kind of an output. So omics, as I said, is a kind of different technique. So that is why it is a very high throughput technique. That's because whatever data is generated when you are doing any kind of omics work is very huge. And it's not possible for any human to study it in that details. So that is why you do require some kind of an tools. So that is why we use computational tools. Now, the data that is generally generated through this high throughput techniques would be, you can have n number of genes that are being generated. You can find out the expressions that you have with regards to genes, various proteins that are being expressed, and also it could be a combination of any two or three, all the three of them. So depending on what kind of study you are doing, that will tell you what kind of data you are getting in. And that data can be then analyzed using computational methods, which is the bioinformatics part, because the bioinformatics is taking into consideration the biology as well as the informatics. It is combining it to give you or helping you to solve certain problems that otherwise would have been difficult, or you can say it could be time consuming at the same end. Say, for instance, you have generated a lot of genes, around 1,000 genes, for instance. So analyzing that genes and seeing which gene is the most apt is very difficult if I say it from human point of view. Using computers, that is what becomes more simplified. So, so omics is basically signifying the measurement of the entire complement of a given level of biological molecules. The information that is obtained is basically encompasses a variety of new technologies, and that helps us to explain how there is a differentiation that might exist between, say, for instance, a normal and an abnormal cell. And when I'm talking about the differences that is there between normal and abnormal cell, I'm also talking in terms of whether there is some kind of a pathway that is affected, whether there is some kind of a gene expression that is getting affected, is there any kind of a mutation that you have come across, or whether everything goes good, but at the end point, when there is the final product that has to be obtained, there is a problem with that. So whatever the place, which we say the 4 omics, all these is what we can study using the tools and the databases that are available for analyzing things. So why do we use bioinformatics? Is obviously because computers is very much helpful and giving us faster results. Along with computers, we also use certain statistical approaches, and that's the best way of studying the variation. And it helps us to perform extensive omics related research that which is present in the biological database, and that helps us to do some kind of comparative studies. Thus, at the end, it helps us to identify sequences of proteins that differ between diseased and healthy tissues. So the branches of omics are, you have genomics, which is nothing but your quantitatively studying of protein coding genes, whether you have regulatory elements and non-coding genes. So these are few things that we study with regards to genomics. Then we have transcriptomics, that is your RNA and gene expression. Then is proteomics, which is focusing more on proteins, and metabolomics, after proteins, whatever we get as a byproduct or as an end product of any kind of a pathway is metabolomics. And that also indirectly helps us to study the various pathways that are involved in that. So it is a clear, you can say very much clear follow up from your central dogma from genes to mRNA to DNA to mRNA to proteins and then to the product. So what can we achieve at the end, or what are the things that we can get after you do any one omics study or a combination of omics studies? You can do data processing. You can identify certain proteins that are being expressed. You can go away with statistical analysis. You do pathway analysis, and at the end, you can also do various type of modeling studies, wherein when I get to know, so target identification is also one thing that can be there when I'm talking about your pathway analysis. So now focusing on individual omics that we have. So when I'm talking about genomics, we always obviously get sequences from various experimental sources, and then they are stored in the databases. Now you have two kinds of databases that are available. You have primary database and you have secondary database. Now how they are different is, when I'm talking about primary database, I simply have done some experiment and that data is stored in the primary database. If I further analyze that data and study as to say what is its functionality, where is it expressed, what is the situation under which it is expressed, all these things, then the information that can be stored would be in the secondary database. From here, then we can retrieve the information. So since it is genomics, we retrieve the sequences that are available over here. And after getting the sequence, we can do various studies. The first thing that you can see over here is we can do sequence similarity. And this sequence similarity, you have it, you can do various types of sequence similarities over here. Okay. So sequence similarity would be, say you are doing pairwise alignment. So so you have sequence similarity, which can be, say, pairwise alignment, wherein I am trying to see whether the same gene from two different species, how far they are different from each other or how close they are to. So what is the one thing that is varying? Say, for instance, if I'm talking about hexokinase, that is present in bacterial genome as well as in, say, a plant genome for that matter. So how are they different? Because the length of the genome is different. Sorry, the gene is different. So what is it that is varying? That is going to help me to understand how there are things that are changing down the line from by evolution. And even though there's a change in the length of the genes that you are getting, though both of them are for the same enzyme, but still the length is different. So what has changed down the evolutionary tree, because of which, even though it is coding for the same particular enzyme, still my sequence length is different? Or what kind of a variation is actually existing? The second type that we can do is we can do multiple sequence alignment, wherein we can actually again compare and find out similar genes or similar enzymes from the same same species. So I have, say, just E. coli. Under E. coli, I have different subgroups that are there. So how these are different from each other? A particular say a particular protein is present in all the sequences, but still what is that is different at the genetic level is what we can do with regards to genomics. And a very frequently used tool that you have is BLAST, that is used for doing that. Or you also have EMBOSS, that is used for your multiple sequence alignment. The next is gene prediction. We get to find out because we already have databases that are stored. So in case when you get some kind of a sequence, say after performing any kind of an experimental work. Now, by doing a searching for those genes that are present in the database, whichever gene we see that there is a similarity that is there, that is one way in which we can find out the genes. On the other hand, we also have many tools that are available, for example, GENEParser or Eugene, which help us to find out which gene it is, what is the functionality of the gene, what is this gene coding for, or is there any kind of a difference that is there in this gene. Say I have got this gene from some kind of a diseased person. So what is it that is different from this person and the normal person for that matter? Any kind of a mutation can be studied using the tool mutation tester. We can study the variation that is there within the species. So n number of different species that are there, we collect the genes of the similar kind, and that's how we can study the variations that are existing. And obviously, we can study the evolutionary relation that is existing among various organisms that are present. Okay, so these are your different types of tools that are available for doing their individual tasks. So we can, at the end, what all things that we can do? We can predict or go ahead and find out biomarkers. We can obtain evolutionary history. We can assess risk management. We can also know about the bioarchaeological, anthropology, evolution, and human migrations. So it's not just restricted to any kind of disease related stuff. You can also go about doing a lot of things over here, like your archaeological survey, anthropological survey, or even for that matter, human migrations. So these are the applications. So you can use the genomic study to find out molecular medicines, and it's very much going to be effective with regards to doing personalized medicine. Then you can go ahead and study microbial genomics. So as in the present scenario, you can see that the gut microbes are said to be of very much playing a very high role in any kind of a disease state. So you can obviously go with the microbial genomics, where you can study the gut microbes in details. Risk management would be with regards to whether you are capable, or whether there are chances of you having a particular disease in the future. So there are certain diseases that are genetic in nature, certain diseases that you have it from due to the environmental stress that is there. So you can definitely go ahead and do genomic studies to find out if there is any kind of risk related to these things. Bioarchaeology, anthropology, evolution, these are again various ways in which we can use the genomic studies. Then you have DNA identification. You can also use it for agriculture, livestock breeding, and bioprocessing. So in short, what I can say is with regards to genomics, it's not just restricted to your disease and non-disease states or things like that, but you can also have its application in various categories. Next, coming to transcriptomics. So that's transcriptomics means now we have the mRNA that is available. So you do have again databases that are there which can be used, where you have the mRNAs that are stored. Now, what you can do first is you can find out the exons that are available, or you can directly use the database that is stored. From there, you can obtain the RNA sequences and convert them to your amino acid sequences. After doing that part, you can do again various kind of studies that are there. You can study the gene regulation studies. So after you get these sequences, you have a lot of genes. So how well are the genes regulated? Say again here, what you're going to actually do is you're going to take it from a set of people who are normal and say a set of people who have some kind of an abnormality or something which is different from others. And accordingly, we can see. Say, for instance, if I just talk about a situation in COVID where we had seen that the Indian population were not that much affected as against the US population or the European population. So we could always do this kind of a study to see what is one thing or two thing that is present in the Indian population or absent in the Indian population, and vice versa with, say, US population or the European population, wherein this kind of a discrepancy is seen. You can, this is just one instance. You can have plants that you can see that they are growing, having the same environmental conditions. They are growing in a particular area, but they're not growing in some other, say, country. So what is it exactly that is affecting? How are the genes getting regulated? Which gene is the one that is being regulated at a higher level or regulated at a lower level? These are the regulatory genes studies that we can do, which can give us some kind of a solution to solve the problem that we are facing. At the same time, you have gene expression studies also. So again, gene regulation, when you're talking, what are the genes that are expressed? So many a times it might be possible that it is expressed, but it doesn't go further. So those kind of studies are also there. We can get to know what are the genes that are upregulated, what are the genes that are downregulated, and that helps us to understand the biochemical reactions that are taking place in the system of one group and the system of the other group. You can observe mutation at the mRNA level, and obviously you can go about doing the evolutionary variation studies that are there. So that will help us in prediction of biomarkers and also help us in searching for new targets that would be helpful for drug development. So various approaches or applications of your transcriptomics approaches are, you can do transcriptome assembly and profiling. You can do small RNA characterization. You can do mapping of the metabolites, protein and transcript profile. You can do population breeding studies, and you can do regulation of gene expressions. So these are the various tools that are available for studying the transcriptomics data that is available, or that we can get it from the various experimental techniques that we have. Next, coming to your proteomics data, again, we get the RNA sequence. From there, we can go about doing the predicting the uniqueness within a family of proteins. We can predict the structure of the protein. We can study evolutionary relationship. We can observe the sequence changes that occurs between normal and disease states. So most of the applications you can see is quite common to all the omics studies that is going to be there. It's just that depending on what data is available to you, you are going to do accordingly. One more thing that we need to keep in mind over here is, as we go down the ladder from, say, genomics to transcriptomics to proteomics to metabolomics, we are narrowing down so it becomes much more reliable data. The observation that we see with genomics data compared to the observation that we see with proteomics data would be very different. Proteomics you can rely more on as compared to genomics data. Okay. So all these things are there. You can always, when you have the proteins, you can always obtain the RNA sequence. And the second is where we always get the 3D structure available in the databases. From there, we can do any kind of an interaction studies. So if we get to know, say from transcriptomics, if we get to know certain genes, that are playing a very important role, or say there are certain genes which are getting upregulated in a particular disease. So those genes become our target. And from there, we can, if the structure is available, well and good, we go through this protein. If the structure is not available, we can do the prediction of structures using the information from the evolutionary relationships or some kind of a uniqueness that is present in that protein family. So this information as well as the domain information help us to predict the structure of the protein. And after getting the structure, we can again go about doing all these studies. Okay, so when we have the sequence itself, we have the structure itself. By doing functional similarity, we can go ahead and do functional predictions. We can predict the domains within a family with regards to the known structure as well as just through the sequence. So at the end, we can find structural changes for the same gene expressed and different environment and tissues. So you have one gene that is coding for say more than one proteins. So we can also see that whether the same gene is expressed at the same level into different environments or not, or what is one environmental conditions, or what is one condition in which it is expressed in tissue X, and in the change in the condition is helping it to be getting expressed in say the tissue Y. So those kind of information again can be gathered through the studies of proteomics. So again, the various applications that we have is you can go for proteome mining, you can go for protein expression profiling, post-translational modification, protein-protein interactional, structural proteomics, protein quantification or differential analysis, and functional proteomics.
Thank you, Dr. Shuchi. Actually, I think we are running out of time. So if you can conclude the session.
Yeah, so these are the various tools that are available. Then you have metabolomics that is helping us at the end. And this is also a very important place where you can get good prediction of markers, because the initial, you can say the samples that are there is not something which is going to be invasive. So that's the importance of metabolomics. You can use just say blood sample, urine sample. These samples are enough, so we do not have to go to too much of ethical issues when we are doing metabolomics as compared to genomics, proteomics, and transcriptomics. So these are the various applications of your metabolomics. So very quickly, I'll just discuss a small work that we had done that would probably help you to understand the omics that is there. So what we had done was we had taken few data sets from your Geo NCPI data set, and we had applied Geo to R to get the information that is there with regards to test and control sample. So our actual work was to sort out the differentially expressed genes which are upregulated and downregulated for Parkinson's disease, and those who are tobacco smokers and those who are not tobacco smokers. Because it was seen through literature that tobacco is helping to delay Parkinson's disease. So we were just trying to see how this is affecting the effect of tobacco is there on the expressed genes. So which are the genes that are being expressed, upregulated, and which are being downregulated. Then those genes we had taken in David, and we had seen what are the different pathways that they are getting affected. So we had obtained around 17 genes that were there. And now we are in the process of finding out new molecules, that are interacting with these, so that we could suggest some new molecules that could be helpful for delaying of the Parkinson's disease. So that's how we can actually go about using the whole omics study, using different bioinformatics applications to get down to the conclusions. Okay. So take home message would be you understand what is omics, you understand you are able to do personalized medicine, which is very much coming in the recent future. You get to know about the different pathways involved and the genes that are expressed. And the whole study is actually very much helpful in prognosis, diagnosis, and treatment of any kind of a disease or conditions. And I would like to acknowledge our director, my colleagues, my students, and my family members for their constant support. And last but not least, I thank the organizers for helping me, for allowing me to giving me this platform actually to share the information that I have. Thank you.